Welcome everyone. I'm Samuel Potter, an undergraduate student at the University of Kentucky, and I'm going to be presenting a project we've been working on. It's population dynamics in live and computational models, and they can be implemented in classrooms ranging from middle schools all the way to introductory college. So this first little cartoon kind of demonstrates where we are in human growth. The human population is quickly expanding, and we haven't really figured out how we're going to deal with this huge increase in the number of people. Our resources are constrained, and we're not really sure how we're going to address those problems, which kind of alludes to the importance of understanding population dynamics in modern times. This next cartoon sort of says the same thing. You can see the 7 billion people representing the number of people on Earth, and seeing how we're really using up our resources, and we haven't really addressed what's going to happen once we've run out of the resources we use. There are many different variables that influence a population's dynamics. In this particular project, we focused on 3 of those variables. The first being the sex ratio of the founding population, that being how many initial males and females were there. The second thing we looked at was the amount of food available. How large can a population grow if they have this amount of food versus this amount? The last thing we looked at was how the size of the enclosure, or the space available, influences how quickly the population can grow. All of these modules have 2 components. There's a live model that uses real live Drosophila in the classroom, and a computational model that uses a computer program to simulate the results you would get if you did it with live Drosophila. Both models are completely outlined in all 3 modules, and I'm going to show you examples of both. Classrooms can either implement one, the other, or both, depending on specific situations. So here's a video showing live flies in a live model. You can see them moving around. Over here is the computational model, and you can see how the real live flies moving around have instead been replaced by these computer simulations of flies, but you can get the same result either way. First, let's discuss Module 1, Population Dynamics and Initial Population Size. So this one's looking at the variable of that sex ratio in the initial population. Our research question is, will a population with only 1 founding female grow less quickly than one founded with 3 females? And our hypothesis, based on what we know about fecundity and the importance of females, is that populations founded with more females will grow more quickly than similar populations founded with fewer females. The live model requires a lot more materials than the computational model would require. You have to have 6 small vials, 6 cotton balls, food to feed the flies. You have to actually have the flies, a means of anesthetizing the flies because you have to sex them. It's important to know which ones are males and which ones are females. That's the same reason you need the magnifying glass to really get in there and look at them. Forceps or a paintbrush to move the flies around, a data table to record your data, sex determining guide, again important in determining which flies are which, a permanent marker to mark on the outside of the vials, and then a computer for the analysis portion. You're probably familiar with Excel, not so familiar with JoinPoint. It's a free software. The link is going to be available on the website. So the procedure for the live model. You have to establish these populations in the 6 different vials, and that's what you're seeing here. You have A, B, and C, which each have 1 male and 1 female, and then D, E, and F, which each have 1 male but 3 females. And you see you have these small vials with the food in the bottom, the cotton balls at the top, preventing the flies from escaping. At this point, the picture you're seeing, the flies have already been sexed, so we've anesthetized them, determined their sex, put the appropriate ones in appropriate vials, and then just set them aside. So now we're going to look at the data collection part. You've already established the populations. This is where you get the data you're going to use. The Drosophila, they get in these vials, they start reproducing, they lay eggs, the eggs turn into larvae, and the larvae turn into pupa. And what we're going to count is pupa. There are the little brown things on the side of the vial. You see on the picture on the left, it's circled with a 1 next to it. After about a week, you're going to start seeing pupa. And at this time, you're going to want to check the vials twice a day, ideally 12 hours apart, but again, ideally. And you're going to want to document the time at which you check them and how many new pupa there are. You're going to mark them so that you don't recount that same pupa the next time. The video on the right was taken several days after the picture on the left. And you can see that the number of pupa has dramatically increased. So counting one pupa isn't that hard. But once you've got, 20, 30, 40 of them, you have to be diligent in marking and making sure you don't recount the same pupa more than once. You're going to collect this data, ideally 12 hours apart every day once you see a pupa, until adults start to emerge. So those pupa are going to turn into adult flies. That's going to be about a week later. You're going to start seeing more adults flying around. That's when you're going to stop collecting data on pupa, and you can just remove the adults, and you're finished with this part of the experiment. You're going to input that data onto an Excel data sheet, which is included for download on the module website. You can see where you're going to input your dates and your times, then how many new pupa were in each vial. And what this is going to do is Excel is going to take that data and turn it into an output that you can then input into another program that's going to do a more sophisticated analysis. And all of these steps are thoroughly outlined on our website. The second program you're going to use is this Join Point program. It's a free download, and you're going to input what Excel gave you, and it's going to do this regression analysis. You see those colored lines? It takes all your data points, and it fits these little lines to them so that you can get an idea of slope at particular points. And slope's important for, rates at how quickly something's happening. So, you've collected the data from this live model, you've put it into Excel, you put it into Join Point, and you've gotten these graphs. How do you draw conclusions? So, we base our conclusions on the graphs we get from Join Point. We look at the one that had one initial female versus the one that had three. Which one grew more quickly, which one had more pupa at the end of the testing period. From the results that we got in the lab, this is our graph from Join Point. You can see the two initial adults, which is one female, grew both less quickly and to a lower population size than the one that had three initial females. So now that we're done with the live model, and the live model takes at least 2 weeks if you're going to set everything up right, and you got to take data every day, come in on the weekend. It can be a burden. So we'll talk about the other method you have, which is a computational model. This one simply requires a computer with the NetLogo modeling software installed. NetLogo is a free download. There'll be a link on the website. And the modeling file, which is the computer program that we designed to go with this module. And again, that's going to be a free download on our website. So here's a video of the computational model. And what students can do is they can set the number of initial females, males, vial size, hours to run. They set the components that they want to test this trial. They hit set up and go, and the model will just run through. You'll see under the output under number of pupa, and it will tell how many pupa were in that trial. Over the course of 5 trials, you can input your data in those boxes on the right, press calculate average, and it'll give you the average for those 5 trials. In this one, it was 21.4. You can clear the average, and then go change it so that there's 3 females instead of 1. Press set up, and you're ready to run for your next 5 trials. This output had a 49. So you're going to continue to do this until you have the 5 trials you want to average. Then you'll be able to compare the average for the 5 trials with 3 initial females to the average of the 5 trials with 1 initial female. So the first module looked at the sex ratios in the initial population. The second module is examining the importance of food available. Our research question is, will a population with more food available grow to a larger size than the one with less available food? Based on prior knowledge, our hypothesis would be that populations with more food will grow to a larger size than similar populations with less food. The live model has a list of required materials. You need 3 large storage containers, about 1 gallon in size, not the big totes, but the ones that sit on a bookshelf. You get them from Walmart or a dollar store. 3 cotton balls, children's modeling material, like Play-Doh, or there's different brands, 3 small jars about the size of a small peanut butter jar, 3 small Petri dishes, and then 3 larger Petri dishes, 3 pieces of filter paper, a digital balance, food for the flies themselves, a way to anesthetize the flies. Again, it's important for sexing them. Magnifying glass, again, for sexing them. Forceps or paintbrush to move them. A small syringe, pipette, or squirt bottle. A data table to record your data. Sex determining guide, and a computer with Excel. Something that's not included on this list, but it's going to be required for it's part of the setup. The teacher's going to need to have a drill or an X-Acto knife or some way of cutting a hole in those plastic containers. So, the procedure. You see where we've cut the hole in the top of that container. The teacher's going to want to have already done that before giving it to the students. Then you take a marker and you mark directly beneath that hole on the lid. So you close the container and put a marker through and you mark on the lid where the spot directly beneath that hole. So now that you've marked the lid, you're going to want to assemble the water apparatus and mark out the modeling material. So the water apparatus just consists of the small jar, you fill it with water, and then you take the large Petri dish lined with filter paper, put it on top of the mouth of the jar, and then invert it. So the water can saturate the filter paper, and it's all contained within the jar and the Petri dish. The modeling material needs to be rolled out, and it needs to line the lid where the container contacts it. The idea is to create a seal that the flies can't get out of. So you just roll it up and then line it around the lid as seen in the picture. Okay, so this is, after you've done that step, you're going to weigh out the food, and it's going to go in a little Petri dish. And you're going to want to put 0.5 gram, 1 gram, and 1.5, like we did in ours. Or you can vary it. You can do 1 gram, 2 grams, and 3. The point is that you have varying amounts of food. And that goes in the little dish, and it's placed on the mark you made in the first step, directly beneath that hole. Then you close the whole container together, and you're going to take more of that Play-Doh, and you're just going to pack it in that lip. The Play-Doh laid down in the previous step creates the initial seal, but you're going to want to really make sure that the flies can't get out. So you're going to take more Play-Doh and just pack it in all the way around to try to make sure that no flies can escape. And the last thing you're going to do is you're going to want to take the cotton ball and plug the hole. So you've created this setup. It's fly-proof all the way around. You have that one hole in the top with the cotton ball in it. You have the water apparatus, and then you have the little dish of food. Of note is that that dish of food has a tendency to dry out. So every 2 to 3 days, through that hole in the top, that port, we would squirt water in the food to moisten it. You can either use, the squirt bottle, syringe, we included them in the materials. And the idea is that to keep the food moist so that the flies, can consume it, but not to flood it because there's also going to be, pupa and larvae in there and you don't want to kill them. After you've created the apparatus, you're going to want to put 1 male and 1 female in each of them. So through the little access port at the top you drilled, you just want to put 1 male and 1 female, probably with tweezers or forceps. And you want to drop them such that they don't fall on the food. If they fall on the food, they have a tendency to get their wings sticky, and then they can't fly around and they may die. So it's important that you avoid the food, but you just drop each of them in. This is the data collection portion of this module. These flies have to die out on their own. So it's kind of a set it and forget it mentality. We created this population, we have everything in there that they need to survive, and we're going to sit them to the side, let it all run its course, and eventually they'll all die from lack of food. It takes between 6 and 8 weeks for all the populations to die out. Once they have, you remove the modeling compound, you open it up, and what you're going to want to do is you want to count the number of dead adults. So there's some in the food dish, there's some on the white interior. You just have to really get down and dirty in it, poke around, and count how many dead adults there are. You'll also notice that it's moist in there, so there's mold that's grown. That's what that green stuff is around the interior. It's harmless, but it doesn't smell very good. Analyzing this data, you're going to input it into the provided Excel spreadsheet, putting how many grams of food, the number of adults to start with, and then the number of adults at the end. And then you're going to use Excel to make bar graphs that you can then interpret. So drawing conclusions based on the graphs that you've made, does the data support or refute the earlier hypothesis? And you can come to that conclusion by comparing the bars for the different trials for the amounts of food there were. Here is our results. That's our actual data. You can see that as the amount of food increased, the number of adults also increased. So more food can support a larger population than less food can. If the live model isn't your thing, which, it's got a lot of setup and it takes a long time to complete, then you can use the computational model. Again, just like earlier, you have to have the NetLogos modeling software and the file that we provide. Here is a video of the computational model. Students can input the initial amount of food in that slider in the upper left-hand corner and the initial number of flies. We use 2, but students can alter that to see if that would have any impact. They press Setup and then Go, and you'll see new flies, which is the number of new flies created from those initial flies, is the output. You can go over and put the output for each trial into the average section over there on the right, and there's one for each of the 5 trials. This one was 27, for example. After you complete it 5 times, and you put your 5 trial data in, you can calculate an average, 27. This will be your amount for, in this case, half a gram of food. You'll then be able to increase the amount of food. We'll do 2.5 grams. Set it up. Let's clear the average first. Set it up, and then go. Now the model is going to run with 2.5 grams of food initially, instead of the half a gram that it had before. That one had 123 adults. This one has 124. And again, it's the same sort of deal. You put your output into that section on the right for each of your 5 trials. At the completion of your 5 trials, you can calculate your average and then compare that to the one from earlier. So that brings us to our third module, which examines the importance of space availability. Our research question is, will a population with more available space grow more quickly than one founded with less space? And our hypothesis is that populations with more space will grow more quickly than similar populations with less space. Required materials for the live model is 3 large vials, and by large, I mean larger than the ones used in Module 1. 3 cotton balls or sponge stoppers. They're going to serve the same purpose they did in Module 1. It's just sometimes the mouths are so big that a cotton ball wouldn't suffice, so you have to have something a little larger. Food for the flies themselves, and again, the means of anesthetizing, magnifying glass, sex-determining guide so that you can determine the sex. Permanent marker, you're going to need your data from Module 1 because it's going to serve as a comparison. If you decide not to do Module 1, then you need to go back and collect some of the data from Module 1 to serve as a comparison. And then you're going to need the same computer programs needed in Module 1, which is Excel and JoinPoint. The procedure for the live model of Module 3 is very similar to the procedure for the live model in Module 1. You establish the populations in these vials, and you have to mark the pupa once they start to emerge twice a day until the first adult comes out. The populations here, however, are all going to have 3 initial females. So instead of having 6, 3 with 1 female and 3 with 3 females, you're going to have 3 vials, each with 3 females. And this is going to serve as a comparison to the small vials in Module 1 that also had 3 females. So now that you've collected the data from Module 3, you're going to input it in the exact same spreadsheet that you used for Module 1 because it's basically counting the same thing, and it's going to convert it into the JoinPoint information. Again, JoinPoint is going to run the same regressions like it did in Module 1, going to give you the same sort of outputs, and you're going to use them the same sort of way. Drawing conclusions, you're going to look at the join point output for the 3 adults and the large vials that you collected in this module, and you're going to compare it from the join point output from Module 1 with 3 females. Here is our results in the live model. You can see that the small vial with 3 females grew a lot slower than the large vial with 3 females. The computational model, just like in the rest of them, you have to have NetLogo installed and you have to have our file. So here you have the computational model for Module 3, examining the importance of available space. So this one's going to have 3 initial females, and you're going to set it up with a small vial at first. Set it up, run it. Just like with all the earlier models, you're going to have that average section on the right where you input your data for the first 5 trials using the small vial. This one had a 65, for example. Run them your 5 times, calculate your average. This one had an average of 61.4. Then you go change the vial size from small to large, set it up, and then complete it 5 more times. Again, inputting your data into the average section. At completion, you'll average the 5 trials, and you can compare that average to the average obtained earlier. So you've seen some different ways of going through these modules. There's the 3 modules, but then also each module has both a live and computational component that you get to pick from. Everything's designed with this mix and match approach. So if you only want to do Module 2, looking at food availability, then you don't have to worry about 1 and 3. If you just want to look at 1, again, doesn't matter. The only caveat is that 3 requires some data from 1. So if you're not going to do Module 1 and you plan on doing Module 3, then you just have to go back and collect that little bit of data from the first one. All of this is designed to be appropriate for middle school students and up. The math required is just a basic understanding of algebra with the slopes, something that most people should get in middle school. The computer models are quickly implemented. We did all the work on this end. So you just have to download them and run them. You could get through all 3 of them in 1 class period. Not something you have to work really long at getting. The live models, however, take a much longer time. Module 2, with food availability, takes up to 2 months for it to run its course. But there are pros and cons to each. Obviously, as far as time is concerned, the computer would be preferred, but the live model is more hands-on. The students can actually see the flies, they can count them with their hands. It's something they can actually see happening in real life. At the end of each module, there are questions to help students draw conclusions, but also build upon what they've already know to predict other things, and that would be really useful as an assessment tool in classrooms. Thanks everyone for listening, and please come to our website listed there, and you can get more details.